Hi everyone, this is an introduction to the Simcyp Static Drug-Drug Interaction Prediction Tool version 1 release 2. The static DDI tool is available as a part of Simcyp Discovery and can be accessed via this button on the toolbar. Additionally, the static DDI tool is available as a standalone tool with identical functionalities. The static DDI prediction tool includes the models outlined in the regulatory guidance from the FDA, EMA, PMDA, NMPA, and now the latest addition, the ICH M12 draft guidance. The main features of the static DDI tool are the basic cutoff models and the mechanistic static model. The results are shown as numbers, and these numbers are compared with the regulator's criteria. The regulator's criteria are outlined in the notes section on the bottom part of the screen. In case the results suggest no risk for DDI across all the regulator's criteria, a green button is shown. And in case the results suggest there is a DDI risk across all the regulator's criteria, a red button is shown. And in case criteria of some of the regulators is met, but results do not satisfy all the regulators, an amber button shall appear. The basic cutoff models allow evaluation if the compound is likely to inhibit or induce metabolic enzymes or transporters at clinically relevant dose and exposure. However, these models do not provide a quantitative prediction of the magnitude of any specific drug-drug interaction. The basic cutoff models tab includes an area for the perpetrator compound parameters, which are used for all of the basic cutoff models: dose, molecular weight, fraction and pound in plasma, and Imax, the maximum concentration in plasma. Below the perpetrator compound parameters, we have separate tabs for basic cutoff models on competitive inhibition, mechanism-based inhibition, transporter inhibition, and induction. The inputs needed for competitive inhibition are Ki and the fraction and pound in incubations. The predictions are calculated with click to predict button. In this case, R1 value suggests no risk for inhibition of hepatic enzymes, whereas according to R1 gut value, we may not exclude the inhibition of intestinal enzymes. The inhibitor parameters needed for mechanism-based inhibition are K-inact, the highest inactivation rate achievable with the inhibitor, and K-ap, the inhibitor concentration resulting in half of the K-inact rate, and finally the fraction and pound in the incubations. The equations used for mechanism-based inhibition differ slightly between the regulators, and this is the reason we calculate R2 and R2-ICH separately for evaluating the inhibition risk in the liver. In addition to inhibitor parameters, the basic cutoff model for mechanism-based inhibition requires the turnover rate constant for the enzyme in question, Kdec, in these equations. These turnover rate constants for different enzymes are shown in the System Parameters tab. Here we have two sets of turnover rate constants. The values in the Simcyp column are based on meta-analysis done by the Simcyp scientists and are used also in the healthy volunteers population in the Simcyp Simulator version 22. The values in the ICH column are taken from the ICH M12 draft guidance. The values can be edited by the user by clicking the padlock open and editing the value, and clicking the padlock closed will revert to the default value. The turnover rate constant of the hepatic enzymes for which the ICH guidance does not suggest a value, such as SUP2A6, the value in ICH column is copied from the Simcyp column. There are separate turnover rate constants for hepatic and intestinal enzymes. And in addition to the named SUP enzymes, there is a placeholder for the user-defined enzyme. Back in the basic cutoff models tab for mechanism-based inhibition, we can select the enzyme these inhibition parameters are referred to and click to predict. The calculation uses the enzyme-specific turnover rate constants, which are visible for the selected enzyme in the system parameter sheet. R2 evaluates the risk for hepatic enzyme inhibition according to FDA, EMA, PMDA, and NMPA guidance and uses the turnover rate constants from the Simcyp column. R2-ICH evaluates the risk for hepatic enzyme inhibition according to ICH draft guidance and uses the turnover rate constants from the ICH column. And R2-CUT is for evaluating intestinal enzyme inhibition according to PMDA and EMA guidance, and this uses intestinal enzyme turnover rate constants from the SimSub column. Transporter inhibition tab includes evaluation of inhibition risk of intestinal efflux transporters, hepatic uptake transporters, and renal transporters. To describe transporter inhibition, we need Ki or IC50 for the transporter and the fraction unbound in the incubations. All the calculations in this tab, like in the other tabs as well, are conducted using the unbound in vitro Ki or IC50. So in case the user wants to use the Ki or IC50 based on total inhibitor concentration, the fraction unbound in incubation should be left as 1. For evaluation of the intestinal efflux transporter inhibition risk, the inhibitor concentration is estimated from the oral dose. Additionally, in case of intravenous dosing, the FDA suggests evaluation based on total maximum concentration in plasma. For evaluation of hepatic transporter inhibition, we need to include a prediction of maximum concentration in the hepatic inlet, and for that we need a few additional input parameters for the inhibitor: fraction absorbed, fraction escaping the intestinal metabolism, absorption rate constant, and blood to plasma concentration ratio. For renal transporters and for hepatic transporters after intravenous administration, the interacting concentration is derived from the unbound Cmax of the inhibitor. And the cutoff values for the inhibitors differ in some cases from regulator to another, and thus it is possible to satisfy some but not all of the regulators. And in such case, an amber traffic light is shown. The inputs for evaluation of induction risk are the maximum induction effect INDmax. Well, the regulatory guidance refer to Emax, whereas the parameter name used in the SimSub tools is INDmax. It should be noted that INDmax of 1 indicates no induction, whereas Emax, as written in the regulatory guidance, of 0 indicates no induction. And then we need int C50, which is the concentration resulting in half maximum induction, and fraction unbound in incubation is used for correction for binding in vitro incubations when applicable. The D factor is a scaling factor which may be used in case there is sufficient data to determine the value. However, in case the scaling factor has not been determined, the regulatory guidance suggests using default value of 1. The mechanistic static model covers competitive inhibition, mechanism-based inhibition, and induction of metabolic enzymes in both gut and liver. And these mechanisms are described with similar parameters in the corresponding basic cutoff models. However, the parameters used in the mechanistic static model and the basic cutoff models are technically not linked in stool. So if you, for example, change the competitive inhibition Ki in this tab, the value in the competitive inhibition basic cutoff model tab will not be affected. Mechanistic static model includes also a static prediction of the perpetrator concentration in the liver and gut wall. And these are calculated from these perpetrator parameters: dose, molecular weight, fraction unbound in plasma, blood to plasma concentration ratio, fraction absorbed, fraction escaping the gut wall metabolism, absorption rate constant, and the maximum concentration in plasma. And in addition to those perpetrator parameters, we need hepatic and intestinal blood flows, which can be found from the system parameters tab on the right-hand side. In addition to perpetrator properties, the mechanistic static model accounts for the contribution of the affected enzyme on the victim elimination in both gut wall and liver. And for that we will need to have Fm, the fraction metabolized in the liver by the affected enzyme, and in case enzyme is present also in the gut wall, the fraction escaping intestinal metabolism. As the mechanistic static model accounts for both enzyme inhibition and induction by the perpetrator, and the contribution of the affected enzyme on elimination of the victim, the results may be taken as a quantitative prediction of magnitude of DDI. However, the predictions of intestinal and hepatic exposure and consequently the prediction of interaction risk tends towards overprediction. Furthermore, the regulatory guidance suggests that for compounds that are both inhibitor and inducer, the evaluation should be done not only as combination of inhibition and induction, but also for inhibition alone and induction alone, as overprediction of inhibition may mask induction and vice versa. Therefore, the static DDI tool reports predicted AUC ratios accounting for inhibition alone, induction alone, and both combined. Additionally, as we have two sets of enzyme turnover constants, two sets of AUC ratios are reported. The first three AUC ratio calculations use the SimSup KDEC values, and the AUC ratios with ICH subscript are calculated using the KDEC from the ICH guidance. And in case the turnover constant for the affected enzyme in the gut is not defined in the system parameter sheet, the enzyme is assumed not to be present in the gut wall, and only interaction taking place in the liver is accounted for in those cases. In addition to the results on screen, there is a possibility to output to Excel file. The Excel output includes a sheet for inputs. Here we have basic cutoff models, mechanistic static model, and the system parameters. And the predictions sheet includes both the prediction results and the cutoff values from the regulatory guidance as a reference. And that's all I have this time. Thank you for taking the time to watch this e-learning video.